Hello everyone, welcome to my presentation. My name is Yuanfang Qiao and I'm from Shanghai Zhongting University. And my presentation focuses on the hydrocephalus region segmentation. The segmentation target is shown in the picture and there are 17 regions, which is interested by the doctors. Due to the influence of Lichi erosion and the brain deformation, we need to design a novel framework to conduct the brain region segmentation in a robust manner. Here is the main framework of our segmentation method, which contains a registration network and a position convolution attention block. In the registration network, there are a dual-pass network to produce more anatomical structural information into the whole brain. Next, we propose a novel positional correlation attention block integrated into the unit. In this block, we design a graph to indicate the relative positional relationship between two brain regions and use the attention layer to refine the feature of different levels in the unit. The ablation study of our mast is shown in the upper right corner, and the depth of different brain regions is shown in the upper left corner. The result shows our PCAB and the registration network can help the unit achieve more accurate and more stable results for the brain region segmentation. As shown in the zoomed block in the green. Our master can locate and segmentation the brain regions where the unit fails to handle. The certificate of our master is available in the GitHub, and it is welcome to test and giving suggestions to our work. Many thanks indeed.
Hello everyone, and thanks for tuning in. My name is Ahmed, and today I'm going to give you a brief presentation about our paper, Dynamic Adaptive Spatio-temporal Graph Convolution for fMRI Modeling. But before getting into the main contribution of this work, I would like to start with the bigger picture and the main research objective of which this work falls in. Put simply, we're trying to find biomarkers for psychiatric disorders in brain activity. And one of the main methods in doing so is using supervised learning, where we try to use the learning algorithm to map brain function activity to diagnostic labels provided by psychiatrists. We use resting state fMRI as our tool to proxy brain activity. However, the main question will always be which input representation and subsequently what learning model architecture to use. A main limitation of some of the common representations used now is that they lack the inductive bias to model brain activity. This creates an even more challenging task for the learning algorithm, especially given the low sample sizes in fMRI datasets. A good inductive bias from our prior knowledge of how the brain works is that it operates as a dynamic network from the microscale of neurons to the macroscale of lesions. Thus, dynamic graphs offer a natural representation to model brain activity, and thereafter, together with the rapid development of geometric deep learning, graph convolution networks offer a promising path to go forward. In creating a brain graph, a natural choice emerges for the graph nodes is ROIs defined by an atlas, and the mean temporal signal of the voxels inside the ROI as the features. However, the main challenge is how to create the edges of the graph. Should we use structural topology? Should we use structural connectivity? Should we choose some similarity measure like Pearson correlation? Such approaches are static and do not capture the dynamic nature of connectivity. Further, these predefined edges are not tailored to a learning objective and could further hinder the learning performance of the model. To this end, we propose to learn the graph structure in an end-to-end manner. First, the graph structure is initialized randomly, and then they are tuned using learnable parameters. We use temporal convolutions for temporal feature extraction, followed by graph convolutions for spatial feature extraction. Together they form blocks, and these blocks are cascaded together for spatio-temporal feature extraction, and then they are followed by dimensionality reduction and classification layer. Our results highlight the importance of a dynamic graph structure in the STGC ends. Further, we show that graph structure is generalizable for the same task across different datasets. Finally, the inferred graph structure can be visualized. However, it's tricky to draw conclusions from dynamic graphs because here we visualize only the graphs of the first layer, and higher order connections could also have an influence on the decision. Thank you for your attention.
Hi, my name is Didem, and I will be talking about our research on how MRI image registration considerably improves convolutional neural network-based disease classification. Machine learning methods, especially CNNs, are used for disease classification like Alzheimer's disease. And the images used in these algorithms usually go through different pre-processing steps, including image registration. Here we investigated the effects of image processing on CNN-based Alzheimer's disease classification. We pre-processed the data using 3 different image registration methods: linear, nonlinear, or no registration but only padding and cropping applied. We used the structural MRI scans from the ADNI dataset. Additionally, we used undersampling to create a balanced dataset for age, sex, and diagnosis. We then created stratified train, validation, and test splits, and in order to get robust results, we did splitting and analysis on 10 different data splits. Image processing, especially image registration here, improves balance accuracy by about 6-7%. However, there is no significant difference between linear and nonlinear registration. Moreover, our results show a strong dependence on the data split. Although CNNs don't need pre-processing and can work well with raw data, our results actually show that here image registration benefits the classification accuracy. It could be due to the fact that Alzheimer's disease-related changes in the brain are rather small, and noise present in the non-registered data is greater. The difference across splits suggests that there are hard to identify and easy to identify cases. Thus, it is important to repeat analysis, especially in case of small data sets. Thank you.
Hi, I'm going to present you very briefly our work on the detection of abnormal folding patterns with unsupervised deep generative models. The human brain is folded, as we can see on these 4 pictures. There is a stability in terms of folding and sulci, but also a high inter-individual variability. Some specific patterns have been correlated to neurodevelopmental disorders, such as epilepsy or autism. Once identified, these specific patterns are very useful, for instance, for diagnosis, but the upstream process of identification is very complex. In this work, our aim is to develop an unsupervised method enabling to uncover cortical folding abnormalities. One of our biggest challenges is the lack of consensual datasets of abnormalities, making the method assessment quite complex. To overcome this, we designed 3 synthetic benchmarks of anomalies. To focus on the folding information, we work with negative casts of the brain obtained from raw MRIs that are called skeletons, and we focus on 3D crops. We then implemented a beta VAE that was trained only on control data. To understand what the model has encoded, we analyzed the latent space thanks to gradient boosting classifiers and generations. Results suggest that our model was able to encode some specificities of the studied region and was able to distinguish benchmark from control subjects. Obvious and finer variations are detected, but the more subtle ones are harder to identify. Interestingly, the main dimension, distinguishing controls from benchmark subjects, could encode the length of the Sylvian fissure. An important limitation of this work is a lack of real clinical data. In the future, we'd like to build benchmarks from neurodevelopmental disorder datasets. Thank you very much for your attention, and I'd be glad to talk with you.
Hello everyone, today I would like to talk about our study, Deep Stacking Networks for Conditional Nonlinear Range of Kai-Cheng Modeling of the Functional MRI Data. In this study, we proposed a method to find out the nonlinear Kai-Cheng network of the text functional MRI data. The conditional range of modeling is defined as the unique inference of one brain region to another brain region after accounting the other source regions. And our proposed model could identify the nonlinear relationship and the time lag relationship between the source and the targets. In the method, we propose to use the deep stacking networks with the convolution neural networks as the components to identify the Granger causality. And the time lag between the source and the target could be accessed to the CNN weighted. And we evaluate our method to the synthetic datasets, simulated datasets, and the real-world functional MRI data. And we compare our results with the multivariate autoregress model and the temporal capture discovery framework. In the results, our proposed model could identify the conditional nonlinear relationship and the time lag relationship in the synthetic datasets and the simulated datasets, where the causality was known as a priori. And our proposed model have outperformed the two competing methods. In the real-world functional MRI datasets, our methods identifies the strong directional relationship between the occipital gyrus and the thalamus. In the conclusions, our proposed DSNCN models to identify the conditional nonlinear Granger causal relationship between multiple time series, as well as estimates the time lags between the source and the target. Thank you for your listening.
Machine learning of task-related and spatially coherent dictionaries from task of MRI data. The goal of this work is to overcome the limitations of traditional voxel-level analysis of task of MRI data. This method uses prior knowledge of temporal pattern of task conditions and locations of brain regions hypothesized to be involved in task. It also decomposes functional MRI data into temporal patterns of signals that loosely differ between task conditions and sparse spatial patterns that are at least partially similar to non-functional network graphs. A baseline sparse dictionary learning method is presented in this work, and the enhancements are made on it to leverage non-task-related time courses. A method to leverage hypothesized threshold half location and spatial coherence is discussed. Experiments are conducted on both synthetic and real MRI data. We use SimTB to generate synthetic task-related MRI datasets, and 15 spatio-temporal patterns were generated as shown in the figure. Coming to real datasets, real fMRI dataset was acquired as a part of a clinical trial among healthy young men in the age range of 18 to 39. And the constrained dictionary learning that we developed does the best job among all the algorithms that we tested in terms of recovering the alerting and orienting related spatial and temporal patterns, even though information about these spatial temporal patterns were not provided to the optimizer. Using the information to partially constrain the dictionary learning allowed this method to identify task-related networks in real and synthetic MRI data. Our future work should assess how additional or other types of constraints help or hurt the performance and how a method can be applied to more novel tasks whose involved networks are less certain.
Welcome to my presentation on structure function mapping via graph neural networks. I'll start with stating the mapping problem. Given an undirected structural connectivity matrix, we use a trained deep learning model to predict the associated functional connectivity matrix. We use the autoencoder as our main model, which consists of an encoder, a decoder, and a loss function. Structural connectivity matrices are processed using Chebyshev polynomials before entering the model. Two encoders were used in our work. They are graph convolutional networks and graph transformer networks. To our knowledge, the original GTN-based framework has never been studied in the context of structure function and brain connectivity mapping. We also demonstrate that a trainable decoder which projects lower dimension vectors onto higher dimensional space can improve the model performance. Our experiments demonstrate that both GCN encoder and GTN encoder can, combined with the proposed decoder, can provide better results on our data than the previously proposed GCN autoencoder model. GTN encoder is also shown to be more effective when it comes to noisy data and outliers. Thank you very much.
Hello, my name is Wenjun from University of Lincoln, UK. It's my great pleasure to be able to present our research at the fourth workshop of machine learning in clinical neuroimaging. The paper I'm presenting today is about multimodal brain segmentation using hyperfused convolutional neural network. Motivated by the recent success achieved using densely connected fusion network, we exploit a new multimodal fusion architecture to overcome some limitations that we identified at feature abstraction and multimodal feature fusion mechanism from the existing network. The proposed architecture is called Hyperfusion Net. It features in gradual increase of filter numbers in the successive CNN blocks, a so-called race fuse module to improve the fusion of multimodal features along each modality path, and use combo loss to benefit from both dice loss and cross-entropy loss. The proposed hyperfusion net is evaluated both on hospital-collected multi-modal data of acute stroke lesion segmentation and on the public ISEG-17 MICA ground challenge dataset. So from the learning curve and the testing results, we can see that hyperfusion net not only become more accurate compared to the baseline, but also converges faster. We also had an ablation study showed the clear improvements made by each proposed modification to baseline network with the race fuse module, making the biggest contribution. From the qualitative results, we observed the proposed network for the application of stroke lesion segmentation is better at discarding outliers and predict a stroke lesion region of higher quality. And for infant brain segmentation using the ISEG-17 data, the proposed network also shows a better contour recovery than that obtained by the baseline. So overall, the experimental results demonstrate the effectiveness of the proposed network and its capability to tackle challenging multimodal segmentation tasks. So that's everything. Thank you very much. And if you have any questions, please don't hesitate to ask me.
Hello. Is it possible for a machine learning algorithm to detect early Parkinsonian patients when it has never seen any? That's what we will see in this collaborative work that we've done with Veronica Munoz Ramirez and involving 3 teams, a statistical team with Florence Ford, an image processing team with Carole Artisien, and a neuroimaging team with Michel Doja. So, how does it work? We will try to train 2 different autoencoders to try to reconstruct only healthy brain images. This one, on top, takes as input full slices of healthy brain images, compresses them into a latent representation, and tries to reconstruct them. We can then hope that when a Parkinsonian patient comes up, the network will not be able to reconstruct the lesions or atrophies present in the brain. This one, at the bottom, takes as input smaller patches of images, compress them and tries to reconstruct them. Also, it is a Siamese neural network, meaning patches that are close in the brain will be enforced to be close in a latent space. We can then get some anomaly maps by computing the reconstruction error. Here are some examples of the reconstructions we get for the 2 networks. So what do you think? Which one looks best? Also, here at the bottom are some of the anomaly maps we get for the controls on top and for the patients at the bottom. So what do you think? Are we able to detect Parkinsonian patients? If you want to know more, come and see the poster.
Defining biologically meaningful cortical regions is paramount to understanding neuropsychiatric disorders. Recently, the Human Connectome Project defined 180 cortical regions per hemisphere using 112 features from multimodal magnetic resonance images. This approach relied on manual annotation of a group template and then propagation of this template to single subjects through a multi-layer perceptron. Despite its state-of-the-art performance, this model was unable to detect 3.4% of cortical regions across all subjects. Deep learning methods have been applied to many medical imaging tasks with exceptional results. As the cortex is best represented as a surface, we assess the performance of 4 geometric deep learning methods on the task of cortical parcellation, since these are adapted to irregular domains such as surfaces. Overall, geometric deep learning methods performed well, with all models achieving a dice overlap ratio of at least 0.85. At a regional level, larger cortical regions had a higher mean and lower standard deviation dice overlap ratio, whilst the opposite was true for smaller cortical regions. As common image segmentation metrics do not fully capture model performance, we assessed whether these models could accurately predict atypical cortical topography in Area 55b. Normally, this is a small contiguous region, but in 10% of people is split into 2 separate parcels. The geometric deep learning models tested here could not accurately predict which subjects had split Area 55b. However, information about atypical Area 55b topography was encoded in vertex-wise uncertainty maps. These results suggest that deep learning methods with uncertainty estimation may be useful for predicting subject-specific cortical organization, which is essential for the precision modeling of neurological and psychiatric disorders.
Hello, my name is Simon, and this is a presentation of our work at the Matrix Lab at King's College London on improving phenotype prediction using long-range spatial-temporal dynamics of functional connectivity. Functional connectivity has been used by many to study the functional organization of the brain, and it has been shown to be hierarchically organized in both space and time. Many deep learning methods apply graph convolutions on sequences of brain connectomes where nodes represent brain regions and edges represented connection between those regions. However, most of those methods separately process spatial and temporal components of fMRI time series. Additionally, network modeling is usually based on group average anatomical or functional parcellation of the brain, missing the individual heterogeneity of cortical topography. To mitigate those issues, our method takes into account subject specificity by building graph sequences using multi-resolution spatial group ICA maps that propagate a group parcellation at the individual level. Then we adopt a deep learning method developed for skeleton-based action recognition, MSG3D, that extracts powerful spatial-temporal features. The model is made of 2 paths, one extracting long-range spatial and temporal dependencies, and the second one applying a newly designed 3D graph operation for spatial-temporal feature aggregation. This method was used on the HCP dataset. We demonstrated that explicit modeling of spatial-temporal dynamics improved prediction of behavioral phenotypes, with improvement of 6.7% on sex classification and consistent improvement on fluid intelligence prediction compared to other methods. Finally, we demonstrate that accounting for inter-subject cortical heterogeneity improves performances while comparing results with models trained only on group average parcellation. Future work will focus on improving network modeling by using full resolution fMRI individual time series. The code is available on GitHub. Thank you for attending this presentation, and please don't hesitate to contact us for more details or questions.